Hi, my name is Chris Bogart. I'm a researcher at Carnegie Mellon University, and I'd like to demonstrate a tool we've been working on called the Scientific Software Network Map. The purpose of the map is to help scientists find out about how software they write is being used. Knowing how often different packages are used and in what combinations can help you decide which packages to spend time maintaining, justify the time you spend on them, and even give you some idea how to enhance them based on how people are using it. So our software map currently collects information about the usage of R packages. Data from the map comes from two sources. First, a set of volunteers who are letting us track which packages they load in each session, and second, an analysis we've done of almost 50,000 GitHub projects that import or require R packages. So let's click Browse and look at some of these packages. There are 5,000 packages from CRAN, Bioconductor, and elsewhere in our database, but I'll focus today on some specialized genetics packages. Let's start by looking at a package called Adejane and try to get a sense of what its role is within the software ecosystem of genetics packages. As you can see, it's not used by any of our volunteers, most of whom are statisticians here at CMU. But we do see some usage here from GitHub projects. There's 20 of them. It's hard to know what that means out of context, so let's broaden the search to all the CRAN genetics packages. Now we're down to 23 packages. They're currently sorted by usage by our monitored volunteers, which isn't very useful in this domain, so let's sort them by the number of GitHub projects that use them. And we can see that it's about the 5th most used one. Another useful way to compare these is to sort by centrality. Centrality tells us not just how often it was used, but considers what it was used with to estimate how central a role it plays among these packages. In this case, the picture doesn't change much. It moves the Adejane up the list a little. Now let's look at its neighborhood by clicking on this centrality number. Now there's a lot going on in this graph, but briefly, the circles are packages, the size is how often they were imported into GitHub projects, and the lines tell you what was used together. So this massive line says that Adejane was used with a whole cluster of things usually. There's more that doesn't fit in the graph, and a longer list is shown over in the bar graph on the right. Another thing to notice about this is the color. So the orange slices here represent use with Adejane, and the blue part represents usage by other projects in other contexts. So this tells you that these big blue packages are actually wider interest general purpose things, not really genetics related at all, like matrix and HTML tools. So let's look at this related package ape, since it has a little more orange than some of the other ones. Now this picture is very different. They're all mostly orange, meaning that they're mostly things that are especially used with ape. These gray arrows, which we couldn't really see before, are static dependencies. So that explains why some of these are orange. Phylobase, for example, has to be used with ape. Other things like Fangorn are independent in principle, but you can see that mostly people use them together. So there's a lot more to look at, but I'll keep it short and just point out a couple links. In the middle bar, there are graphs and lists of the actual GitHub projects that use the package you're looking at. On the far left, you have a survey link, which you can follow if you want to give us some feedback after taking a look around. And then these about links tell you details about how the data was collected and what our research is all about. And then finally, if you click on About the Data and then Volunteers, you can let us collect your own usage data and help us expand that part of our data set. You install a package, consent to the data being sent, and you don't have to worry about it again. So that's the quick tour. I've put my email here in this video if you have any questions, bug reports, or suggestions about what would make this more useful. Thanks.